Hi everyone. Today we're going to explore a very important topic. Can artificial intelligence and machine learning be used to predict drug safety through what is known as absorption, distribution, metabolism, excretion, and toxicity modeling? To guide our discussion, we'll take a look at a recent review article titled Computational Toxicology in Drug Discovery. I'll also share some of my own thoughts and opinions along the way. If you're curious about the details, I highly recommend reading the full article. It's open access and packed with helpful references to predictive tools, software packages, and web servers. The drug discovery and development process typically takes 12 to 15 years from initial disease understanding to market approval. It is extremely costly, often requiring $1 to $2 billion or euros, and has a very high failure rate, as over 90% of drug candidates never reach the market. Because patents usually last 20 years from the date of filing, and around 10 to 12 years are spent in research, preclinical, and clinical development, companies often have only 8 to 10 years of effective market exclusivity before generic competition can begin. It is important to note that timelines, costs, and success rates can vary depending on the type of therapeutic agent. What we see here is more for small chemical compounds and peptides. And the cost of treatment for patients also varies widely depending on the type of therapeutic agent and many other parameters. Small molecule drugs, like most oral medications, are generally less expensive to produce and purchase. Biologics like monoclonal antibodies tend to be more costly due to complex manufacturing processes involving living cells and stricter handling requirements. Gene and cell therapies are currently among the most expensive treatments, often costing hundreds of thousands to over $1 million or euros per patient. In light of the growing financial strain on healthcare systems worldwide, the current trend of prioritizing the development of mostly high-cost biologics needs some thinking, especially in cases where small molecules or peptides could offer effective alternatives. Absorption, distribution, metabolism, excretion, and toxicity are 5 key properties that must be optimized during drug discovery and development. Roughly 20% of drug candidates fail due to such issues, while around 60% fail because of poor therapeutic target selection. Thus, while safety problems are complex, choosing the right biological target or pathway is also extremely challenging. Now, I'd like to deviate from the review article for a moment and share some observations from my experience in academic drug discovery. For the past 30 years or so, we've been hearing the big data mantra across the biomedical field. Billions of euros have been invested in large-scale data initiatives, and today we indeed have vast amounts of omics and bioimaging data. One problem is that these data consists most of the time of correlation matrices, rich in statistical associations, but offering limited causal information to drive critical decisions in drug discovery. When it comes to absorption, distribution, metabolism, excretion, and toxicity, data remains scarce, most often fragmented, and in general, severely underfunded. We are nowhere near achieving big data or high-quality data in this domain, because generating such data usually lacks immediate appeal to those setting research priorities or allocating grants. It often receives minimal attention and support. But without it, real progress in predictive toxicology will remain slow, at least in my opinion. Let's return to the review article now. The paper presents many tools that can be used for absorption, distribution, metabolism, excretion, and toxicity predictions. I believe it is easier for our discussion here to group computational methods based on their strategic role in early-stage drug discovery rather than by algorithmic type. Following this logic, I suggest to classify methods into 3 main categories. First, we have rule-based filtering methods, which serves as a practical guideline for simple compound triage. These are first-line filters designed to quickly flag compounds with potentially poor drug-like properties before detailed modeling. And again, these are crude indications that may not apply to a specific project, disease, compound families. These filters rely on empirical rules or structural alerts such as the Lipinski's rule of 5 for estimating oral absorption, the search for pains or pan-assay interference compounds, and the detection of toxicophores. These filtering tools are not predictive models in a strict sense. They just provide a crucial initial overview. So all these rules are best used as guidelines, not dogmas, flagging compounds for careful consideration rather than automatic rejection. Second, we have specific endpoint predictions. Here the goal is to predict endpoints such as cardiotoxicity, hepatotoxicity, or metabolism, among many others. These models employ classical machine learning or deep learning methods and can even use 3D protein structural data as input. Deep learning does not always outperform classical machine learning in absorption, distribution, metabolism, excretion, and toxicity predictions because most datasets are small. In such regimes, deep models tend to overfit and may generalize poorly unless techniques like pre-training, transfer learning, or data augmentation are used. In real-life projects, simple classical machine learning approaches often turn out to be more data efficient and interpretable, although deep learning workflows are always worth exploring. Third, we have off-target prediction. This category focuses on predicting binding to receptors or enzymes outside the intended therapeutic pathway, which may trigger unwanted side effects. Different strategies are applied, ranging from similarity searches to highly complex ligand target prediction models. Such advanced predictive engines may use as inputs protein language models for the targets and molecular graphs for the ligands to predict if a compound could bind to a sequence from the human proteome. Yet, given the complexity, these predictions unfortunately do not achieve high accuracy, but still can give some insights and should be combined with other approaches. The review article then lists several online tools that researchers can explore. Among them is FAF Drugs, a platform our team has been developing since 2005. It helps filter chemical compounds based on simple empirical rules and has been widely used by research groups around the world. We are currently working to optimize the tool by refining some of its computations and improving the output diagrams. The review then moves on to present and explain a range of databases, including chemical and toxicity databases, environmental toxicology resources, and biological toxin repositories. Next, it outlines methodological workflows for drug toxicity prediction and introduces tools designed to assess specific endpoints such as cardiotoxicity, hepatotoxicity, nephrotoxicity, acute toxicity, and carcinogenicity. Finally, the authors dedicate a section to how drug toxicology research is evolving in the era of large language models. Present in silico filters and prediction engines in the field can certainly provide valuable insights, but for many critical endpoints, the predictions remain weak. This isn't because the algorithms are poor, it's because the underlying data are missing, incomplete, or unreliable. Unfortunately, very little funding is directed toward generating high-quality data in this field in academic settings. For now, it's not the kind of flashy research that attracts much attention, but without it, real progress in predictive toxicology will remain slow. The discussed article provides an excellent overview of the field. Some aspects, such as drug-drug interaction prediction, are not covered, but including them would likely have made the article too long. I hope you found this very simple introduction about drug safety both clear and useful. Until next time, take care.